Okay. Share my screen. Okay. All right. So the supplies that we needed for yesterday's lab have arrived, or Monday's lab have arrived. So when we gather on Friday to do our lab experiment, we'll combine what we were going to do Monday and then what's on the syllabus for Friday. So that's group one that will meet in lab on Friday. And we're going to continue on a little bit with just some basic ideas on habitats. And then I think on the syllabus, I'm going to jump ahead a little bit to methods in sampling after that. Since I'm falling behind, I will fit the other stuff in on populations and diversity as we go. So I'll just weave that into some of these other lectures. But some general things that I wanted to say on habitats. So freshwater, of course, we have a lot of freshwater here in the Midwest. We're very lucky that way. And if you start thinking about who's living in the Great Lakes, in the upper reaches anyways, we have phototrophs, of course, because they have nice, easy access to sunlight near the top layers of the water. And when you get into the very top, and where oxygen is more abundant in the upper reaches of the water column, you will have, of course, the ones that can carry out oxygenic photosynthesis. So that's the cyanobacteria and the algae. And there's plenty of single-celled algae out there, especially here in the freshwater area, and well, in the oceans too. And then, of course, when you get into some of the other areas of the ocean, there's the kelps and things like that that also do photosynthesis. But as you start to go down into deeper areas of water, you can get into pretty anoxic conditions. And oxygen just doesn't diffuse through water very well. It's a very slow process, and it doesn't reach as far as you think. And so you can get anoxic conditions in the water column. But we already know that there's plenty of phototrophs that can do anoxygenic photosynthesis. So they're there and cooking along. And of course, they're considered primary producers because they're photosynthetic and they're making their own sugars. So they're at the base of the food web and are incredibly important as food for other organisms, as well as the activities that they do by providing oxygen to the rest of us who need it. So again, oxygen, not the greatest solubility in water. And so when you get into deeper reaches, you're going to have lack of oxygen. And then of course, when you get way down deep, nothing going on there at all. But that doesn't mean it's the end of the world, because we know that there's plenty of microbes that can take or leave oxygen. If they don't have oxygen available, then they could do fermentation, they could do anaerobic metabolism. And so to be facultative means you can, you have that flexibility. If oxygen's available, great, use it. If it's not, you can switch to some other method. And so facultative would be my choice. If I had to be a microbe, I'd want to be facultative. So, and then as you get down into anoxic conditions, of course, there are many microbes that don't want oxygen. They have adapted into environments where oxygen just really isn't there consistently. And so they're anaerobes by the selection pressure of their habitat. And then, so then you have obligate anaerobes that die in the presence of oxygen if they're exposed for too long. And then you have kind of, there's the facultative anaerobe idea as well, where oxygen may be present and they can use it. And if not, they'll switch. And then microaerophiles, I think I mentioned them briefly, they don't like 21% oxygen. That's what we're breathing. They prefer usually 10, 11% is like the general number that gets thrown out. in terms of what the microaerophiles prefer. So they still use oxygen, just at lower levels. They can't be exposed to full 21% for too long. So they have some interesting physiology and needs in that way. And then, of course, most of Earth is covered by water, and most is ocean, marine systems. And so generally, when you think about where microbes are, we kind of split it into regions of the ocean. We think about what's happening along the coastline of the land masses, because of course what's happening on the land has profound influence on what's going on in the ocean, particularly the coastal areas. And as you think about all the things that run off from terrestrial environments into the ocean, from a microbe perspective, a lot more nutrients available as you have the coastal interaction with the ocean. So definitely more nutrients. Along the coastal area, you also see high populations of photosynthetic organisms, and just in general, more of everything, because there's just more nutrients. And so they have higher population numbers along the coastlines. And if you looked at any aerial maps, I have an aerial map here. And so here they're showing the Great Lakes in the upper left area. But what I want you to look at is the dotted line that they've drawn here. That's the coast. And then all of the dark red area right off the coastline, that is microbes that are showing up. And so here you can see on the East Coast, you have quite a bit of microbes right along the coastline. And of course, current action and disturbances like hurricanes and things like that will obviously influence this, but in general, it's pretty consistent. You have lots of microbes along the coastal regions.
Wait, quick question.
Yeah.
So the dark area was microbes?
Yeah.
Or is that less microbes?
More. So this dark pink area,
Oh,
all of that along, because the dotted line is the actual coastline.
Okay.
And so they were able to capture that image where they could show plenty of microbes along a coastline like that. I don't know if they say in the notes how they, so that pink is what they did was picked up on the chlorophyll using satellite imagery. The east coast of the US from mid-Florida to northern Maine is shown in dotted outline. Areas rich in phytoplankton are shown in red. Blue and purple have lower areas of chlorophyll. So they were zooming in mostly on the phototrophs. But of course, that's not the only organism along the shoreline. There's other microbes there too. Now, if you get out into the open ocean, there's a chat message. Whoops. When you get into the the open ocean, it's a little bit different story. So that's weird. Why is there such a drastic change between coastal microbes in deep sea areas? Well, that satellite image was looking at just photosynthetic. Let me go back. So they zoomed in on chlorophyll. And so you can see there's some purpley areas here in the blue. That's like the next level of population. And then the blue is probably the lowest population. Yeah, and there is a very sharp demarcation here. But yeah, I don't have an easy answer for that. I don't know how sensitive this satellite is when it captured the chlorophyll concentration. So I'm sure there are other organisms here, and you can see in the purpley areas for sure. Yeah, but it is pretty sharp, I agree. But I think it has to do a lot with the equipment that they use, how sensitive it can be. and they only zoomed in on chlorophyll. There's plenty of other microbes there that they didn't capture. So I think that's part of the reason why you see a sharp demarcation. In the blue region, I think there are other microbes there, but they're just, and yeah, certainly. And I don't know on the satellite, like which chlorophyll they zoomed in on, because there's chlorophyll A, B, C, D, and then there's bacteria chlorophyll. And, you know, so it just depends on how broad, I guess, they targeted the chlorophyll. Open ocean, generally, a good rule of thumb, nutrients are limiting compared to coastal areas for sure. And in particular, nitrogen, phosphorus, and iron are limiting. Now, billions of years ago, iron was super abundant in the ocean, but now it's become much more limiting. And so when you think about who are there, it's photosynthetic. Obviously, that makes a lot of sense to have them in the upper reaches of the open ocean where sunlight is beaming down on the ocean with no shading from trees or anything like that. And so they generally say the top 300 meters are where you're going to find phototrophs that can do photosynthesis. Whoops. And then as you get below that, because sunlight doesn't penetrate down, well, and then there's different qualities of light that will penetrate to different depths. And so like red light can penetrate deeper than, you know, some other qualities, for example. And so that's also why you get certain, of course, we talked about that before in the water column, you can have many different types of microbes that are doing photosynthesis, and they're capturing different wavelengths of light depending on their pigments and what their pigments have adapted to. And so, you know, when you think about the rainbow of colors, the quality of lights, red, orange, yellow, green, blue, indigo, violet, you do see that those wavelengths will penetrate to different depths. And the microbes can capitalize on that. They can adapt depending on their pigments. Again, just trying to get you to think about general population levels. They usually say, you know, 1 times 10 to the 5th to 1 times 10 to the 6th cells per mil is a general, you know, range to think about. Of course, there's huge variation on that depending on where you're sampling out in the ocean, but this, we're talking about in the upper reaches now of open ocean. You get around that range. And then as you sample deeper and deeper, you're going to get progressively less microbes. But so, for example, when they've taken samples around 1,000 meters, you get, you know, 10 to the 3 to 10 to the 5 cells per mil. Then, you know, they've taken samples in pretty much every water, you know, ocean system on Earth, and again, some general ideas you can think about. The domain bacteria, the organisms that belong to that domain, they tend to dominate in the upper reaches of the ocean. They're better adapted to that. And then as you go deeper, it tends to switch, where domain archaea type organisms tend to be better adapted and have higher populations. It's not perfect, I'll show you an image, but that's a general rule of thumb. I would guess most of those are in the upper reaches, right? Most fungi. Yeah, that's a good question. Yes. But the deeper ocean, I'd have to look into that. I haven't run across any papers where people are studying the deep ocean for that, but I could just be missing it because I, you know, haven't Googled the right search term. So yeah, and the textbooks where I'm getting this information from, they kind of ignore fungi too, which is sad. They tend to talk about the bacteria much more. So here is some huge numbers here. 3.1 times 10 to the 28. So that's, you know, 28 zeros following 3.1. So this is definitely an estimate, but this is common. I've seen these numbers in published literature and in textbooks. where they're looking at, globally in the oceans, what do we think we have? And the bacteria and archaea tend to dominate. There are domain eukarya organisms there too, but when it seems to be heavy on the domains bacteria and archaea that make up these numbers. And so when you think about the oceans in total, most microbiologists would agree the data is showing that you have the largest microbial biomass on Earth in the ocean. And so obviously profound impact on, their activities have a profound impact on cycling of nutrients and atmospheric gas composition. So they have, the ocean systems haven't really been heavily studied for microbes until relatively recently, when technology has gotten better and sampling techniques have gotten better, and also being able to sequence DNA fairly cheaply. Probably, well, the most notable person that has gone into the ocean system his name just flew out of my head. He was the businessman who owned the company that helped sequence the human genome. Oh my gosh, I'm blanking on his name. I just had it. Anyways, after his business and then several other labs sequenced the human genome, and then after he did that, he went into the ocean and said, let's go mine the ocean for interesting drug discovery. And initially his idea was drugs to, especially to fight cancer and things like that. And then he started realizing that there's all kinds of other microbes in the oceans that can be used for all kinds of things, not just cancer drug discovery. So he has gotten way into algae, and he is saying that we should be farming algae for food production to help feed 8 billion people almost. And so he is spending a lot of money looking at these ideas. So he's busy mining the ocean for these types of ideas. And I'm still blanking on his name. I'm waiting for it to pop into my head. We'll get there eventually. This is just a visual trying to show you the water column in the ocean. And so you have depth on the y-axis here on the left, and you see they took it past 5,000 meters, and you can see how on the y-axis it's percent of total count. So they just looked at domain bacteria and archaea, and you can see that the bacteria win as you're at the upper reaches. There's more total percent of bacteria compared to archaea, but then you can see that there's this inversion, so to speak, where you have the archaea starting to dominate. But it's not huge, it's not super dramatic, but you can see at the deeper reaches, the archaea are better adapted. At least there's more species that seem to be better adapted and can dominate to some extent in the deeper depths. And then they're just showing counts on the far right. So that's a nice visual to get you thinking about some general rules of thumb, at least with those two domains. Okay, so often when they've been sampling, it is fun to think about, of course, one of the early questions was, are microbes truly everywhere? Are we going to find some habitat where they just can't survive? And so far, it doesn't seem to be the case. It seems like no matter where we look, there's microbes of some sort, and some are just fantastically well adapted to what we would consider very bizarre or hostile environments that there's no way we could survive, and yet they have, through natural selection and adaptation, evolved really interesting techniques to survive some bizarre areas. So this table is getting at some of the sampling that they've been doing globally, and you can see that there's some terms here showing up on the far left column. So hyperthermophile, hyper, well above. Thermophile is referring to heat. And phile, if you're a P-H-I-L-E, if you're a phile, like if you're an audiophile, that means you love music. So phile is to love. And so hyperthermophile, they love really hot conditions, 121 degrees Celsius. I've talked to you very briefly about 121 degrees. There's a piece of equipment we use that's set at 121 degrees. What? The autoclave. That's what we use to kill most microbes is 121 degrees. And here they're saying, well, there's some species that can handle that. So if we popped pyrodictium into the autoclave, it would probably survive. And but in general, we don't have pyrodictium living in, at surface areas outside of the ocean. They're in the hydrothermal vents. So in fact, that's one of the problems when they are sampling deep ocean or any of these really bizarre areas. Actually, when you take a sample from there, it's challenging to not kill the microbes because you take them out of their environment into such an extreme different environment here at the surface or in the lab that often they can't handle that and they die. And so that has been a huge challenge. It's how do we keep these microbes alive when we bring them to the surface, for example, and such, we have to store them and treat them in a way that keeps them alive. So it's been pretty challenging. And then let's see, psychrophile is the opposite idea. Psychro is cold, and so you love cold. So when they say phile in microbiology, what they mean is if you're a psychrophile, you have actually adapted to that. It's not that you can just be exposed to it and survive. You actually prefer. So what they mean is you've adapted to that type of really cold condition. And so negative 40 degrees Celsius, can you imagine? I mean, we can't do that. Lake Vostok beneath the Antarctic ice sheet. So that's another example. And then you have high acid conditions, acidophiles. From acid mine drainage, unfortunately, there's plenty of acid mine drainage environments. Probably one of the most famous is on Iron Mountain, which is kind of weird because we have Iron Mountain here in the UP, but there's Iron Mountain, California. They have, unfortunately, a well-studied acid mine drainage system there, but pH 0.5. What's our stomach? Our stomach acid is what, pH 2, I think, between 2 and 3 maybe. That's about as acidic as we get. That's just part of our body. Halophile. If you're a halophile, that means you love salt. And hypersaline basin, for example, in the Mediterranean. So super salty conditions, which would, when you salt your food, one of the reasons your food stays fresher, if you like to preserve fish, for example, they often salt the fish for storage. And that's because you're killing most of the microbes. Salting creates such a salty environment, such a saline environment that it literally sucks the water out of any microbes that are hanging out on the fish and they die. So that's why salting can preserve food so well. Alkaline conditions, alkalophile, that means you like high pHs, so pH better than 10 here, soda lakes. So as you're looking through this, the terms I'm going to be asking about, so please start memorizing those terms and understanding what that means in general. Now, I won't say, give me the exact temperature that a hypothermophile likes, but if you can explain to me in sentence, just the concept of what a hypothermophile is. Numbers, I'm not too worried that you memorize, the range of numbers because it can shift as we discover more and more. So I'd rather you're just comfortable with the general concepts. Let's see, what else? Radiation tolerance. Here's a nuclear reactor water core. Can you imagine? Deinococcus, I remember when that was discovered. Deinococcus radiodurans is a bacteria that they found in a nuclear reactor water core and was happily living there, well adapted to those conditions. So just incredible. Of course, they've been studying that quite a bit because it's like, wow, it can handle that level of radiation. What is it doing? And maybe we could learn from that and help. I could help with radiation exposure. Okay, barophile is another one. I tend to use barophile, but depending on who's talking or what you're reading, they might say piezophile. It means pretty much the same thing. High pressure. And so as you get down, if anyone has done scuba diving, you're familiar with that idea that as you get deeper and deeper, the pressure increases on your body and you have to be careful about that. as you ascend back to the surface. And so they're well adapted to barophiles. And that was another problem. When they would go and sample in the deep sea, they'd bring up the microbes, they grab a sample and bring them up, and they just pretty much explode when they got to the surface because they needed to be under pressure the whole time. So it's been very challenging. Oligotroph just means that they're used to low nutrient concentrations, and they have special ways of dealing with that. Xerophile is another term I'd like you to know. Really consistently dry conditions, lack of water, and so they have been able to adapt to very sparse water. And you can see fungi fit into that. That's one reason, if you've ever, what tends to happen a lot with me with my food is I'll buy some jam or jelly and use, open it and use it, and then I'll put it in the fridge and forget about it, and then I'll take it out again and there's a fuzz of fungi growing on the surface. I don't know how many times that's happened. And so really jam and jelly is technically pretty dry because it's so sugary. You're creating a situation where actually free water is difficult for most organisms, but fungi, they're good at it. So that's why you tend to see fungi on your jams and jellies. And then endolith, endo within, lith, rock. And so surprisingly, they have looked at, even, well, we've already talked about deep into the ocean core, you can see microbes. But for example, in Antarctica, where the weather is so extreme, microbes tend to go into the rock in order to survive the extreme conditions. And so when you do some rock sampling and bring them back to the lab, you'll find plenty of microbes in the rock hanging out because it's a nice sheltered environment. So these terms I'd like you to think about and then be able to give me just a general example of what kind of habitat are we talking about? Where would we find these types of microbes? So the first two columns in this slide and then in this slide, please be ready to think about that and talk about it on the next exam. Well, let me give you a few examples of some of these extreme organisms and how they're adapting. So I'll give you a few ideas here. So if we go deep down into the ocean, we're looking at the below 1,000, typically is where they say it's deep sea. Well, it's very cold. If you've done any scuba diving, even here in the Great Lakes where we go just, 100 feet or so, you can certainly tell the difference between the temperature deeper down versus the ocean or the water surface here in Lake Superior. And then in general, you have low nutrient concentration in general, except near hydrothermal vents. Hydrothermal vents often have plenty of nutrients. So it's patchy when you get down into these depths. Yes, there are certain select regions where you'll have a flush of nutrients, and we'll talk about that. But in areas away from the hydrothermal vents, just at 1,000 meters in the water column, for example, low nutrients is pretty much the norm. And so psychrophiles really adapted to nice cold conditions. And not only are there psychrophiles, but plenty of them are barophiles too, in the sense that they can handle pressure. And so if you've taken scuba, they always teach you pressure increases by 1 atmosphere for every 10 meters of depth that you go down. And so you can have, depending on the water column and what the pressure is, you'll have some that are called barotolerant, which just means they can handle that, but they're not really well adapted, and so they tolerate it. But often you will see that when you get way deep, then you get into the philic condition, the barophilic or barophiles. If you're barophilic, that just is more of an adjective where you're thinking about that you've actually adapted now to those higher atmospheres. And then, so that's about 4,000 to 6,000. So barotolerant is above that, barophilic, and then the extreme barophiles down 10,000 meters in the ocean, that they're hanging out there and they've adapted to those extreme pressures. And again, trying to sample those, it's been really challenging because you bring them out of that amount of pressure, and if you don't keep them under pressure, they're going to die. And of course, it's pretty difficult to study them then if you can't have them living. So this is just a visual of what we were just looking at, showing the general trade-off here, where you have the barotolerant, where you can see they're more adapted to the upper reaches, but you know, they're hanging in there until you get around 500 or so atmospheres and their populations totally go extinct. Barophile, they're showing they don't do so well in the upper reaches, but they really are happy in terms of being able to stay alive and reproduce at more you know, higher atmospheres of pressure, certainly, than the tolerant, and then the extreme barophile. So, of course, we wanted to know what are, especially the extreme barophiles, that they're fun to think about. What are they doing to deal with this kind of pressure? And so when they finally figured out how to keep them alive after they brought them up to the surface and started looking at their cells, just their morphology, and then the composition of their, especially their cell membrane, because of course the cell membrane is incredibly important for, well, any organism, but particularly for the microbes, because they don't have internal organelles to put their proteins and enzymes in those. So when they looked at their cell membrane, what they found was there were nice high levels of the unsaturated fatty acids showing up in the cell membrane. So when you look at our lipid bilayer membranes, they're composed of glycerol and fatty acid tails, and there's different fatty acids. And I think if you've read anything or listened to health experts, you know, we talk about unsaturated fatty acids, monounsaturated fatty acids, polyunsaturated, and then you have, of course, you have the saturated, and how, you know, different fatty acids are better or worse for our health. And so it's the same idea here, that you have a variety of different fatty acids that can make up the cell membrane. And microbes are good at switching out fatty acids as they need to in order to adapt to their particular habitat. So with the barophiles, with the barophiles, especially the extreme barophiles, you see this really high level of unsaturated fatty acids. So hang on, well, let me see if I have that. Where is it? Hang on. So let's go to this picture. So here is a lipid bilayer membrane. So all the brown squigglies, those are the fatty acids. The blue spheres are the glycerol molecules. And so, and then up top you have kind of a blow up of that. So you have the glycerol and then the fatty acid tails that hang off of the glycerol molecule. So that those fatty acids can vary. And so when you look at here, you see the actual chemical composition of the fatty acid tails, all right? And then let me show you, this one is just showing some options that you can have when microbes are building their cell membranes. They can build different fatty acids into their cell membranes depending on their habitat. So here on the left, we're looking at saturated fatty acids, which means there's no double bonds between the carbons. These are carbon-to-carbon bonds. So if there's, this example is also fully saturated. You have no double bonds showing up between the carbon. In the unsaturated, you have, in this particular image, it looks like you have a little bit of difference. Here you have one double bond, so it's mono unsaturated. Here you have, if it's more than one, then they switch to polyunsaturated, all right? But it can vary. It can be 2 double bonds, 3 double bonds. And what do you notice? So look on the fatty acid tails on the left, and look how they drew it, you know, that kind of up, down, up, down, up, down. What do you see over here on the right? Yeah, right? It's switched to being, it's not so nice and symmetrical as what you see with the saturated. It's got these weird kinks and bends, right? Those are the double bonds causing that. So when you have a double bond in between the 2 carbon atoms, it tends to kink the tail, for lack of a better description. It tends to kink or bend the tail. Well, then if you can imagine these bent tails, fatty acid tails, they don't lie next to each other. I always get that wrong. They don't pack next to each other very nicely because of all those bends and kinks. So here with the saturated, they can sit next to each other quite nicely. But if you have double bonds, they don't pack very nicely next to each other. Well, that actually has a profound influence on whether or not that membrane will be fluid or more gel-like. And so if you can imagine when you have unsaturated fatty acid tails and a lot of them, and then you start applying pressure, like, deep in the ocean, you start applying pressure to that cell membrane, that's actually a protective mechanism. And I often use the analogy, think about butter versus olive oil. So if you have a stick of butter in a butter dish and you leave it out on the counter, even when it's 80 degrees in your house, does it liquefy into total liquid? No. Is butter composed more of saturated or unsaturated fats? Saturated. So that means butter has more of this idea where you have a bunch of saturated fatty acids. They pack well together, which means they gel up and they can stay solid at room temp. Even if it's 80 degrees, the butter stays relatively solid. And that's because of the fatty acid tails being able to pack next to each other under cooler conditions. On the other hand, olive oil is liquid, in your cupboard. That's because it has, mono and poly unsaturated fatty acid tails don't pack together. So even at 80 degrees, or, well, I should say, even if you stick olive oil in the refrigerator, it still stays liquid, if you've ever tried that. Well, it's the same idea with a barophile. If you put a lot of pressure onto the cell membrane, if they have a lot of polyunsaturated fatty acids, those fatty acid tails don't line up very well with each other, even under pressure. And so that keeps their cell membrane more fluid. That's really important. You need to have a fluid membrane in order for it to function correctly, and for transport to happen across the membrane, and for the proteins embedded in the membrane to function correctly. So having a relatively fluid membrane is incredibly important for any living organism. And so this is one strategy that they have done to really adapt to these high pressures. They just pack their cell membrane full of lots of unsaturated and probably polyunsaturated fatty acids. Some other things that they've noted with these barophiles is that their membrane transport proteins are different. Their physical structure and composition of amino acids are different when they are in these high pressure environments. How protein synthesis occurs is a little different. I'm not going to get into it. The protein structure, including how of course, we know proteins are just a bunch of amino acids bonded together, but then don't forget they have to fold. That chain of amino acids has to fold into a particular three-dimensional shape. And if it doesn't fold correctly, a lot of bad things can happen, including some pretty terrible diseases that impact us. And so they noticed that the proteins fold differently into different three-dimensional shapes when they're at those depths, but it's a way of them to adapt to these very high pressure environments. The trade-off of living under such high pressure is a slow growth rate. These microbes have a much slower growth rate, and that's a pretty good rule of thumb. Most extremophiles in general, no matter what the extreme condition is, they tend to have a slow growth rate. So that's definitely a trade-off, but at least they can survive. Okay, so that's really interesting. Probably the cell membrane has been best studied, but they're getting into some of these other ideas as well. So here on this table, they're looking at, okay, how are they adapting to some of these other extreme conditions? So cold is similar in some ways to high pressure. You're still going to see lots of unsaturated fatty acids. Think about olive oil in the refrigerator. If you have lots of unsaturated fatty acids, you're still going to stay liquid under cold conditions, or fluid, I should say. And so, and then the enzymes just are really quite capable of being functional under cold conditions where, you know, our enzymes certainly aren't. If we get too cold, we die. Heat. So membranes, on the other hand, if you have high heat conditions, you're going to have a lot of saturated fatty acids to pack your fatty acid tails together, so that the heat doesn't cause your fatty acids to totally fall apart from each other. You're pretty well gelled up in some way of thinking about it. So again, thinking about what are some of the challenges in terms of the environmental extreme, and being able to give me some examples of what they would do. So this table, you know, thinking about what could they do in order to deal with these extreme conditions. So please spend some time looking at this. The high salt idea, if you are in a really salty environment, and there's plenty of microbes that have totally adapted to that, they will either make themselves really salty, so to speak. They'll pack a lot of solutes inside themselves in order to keep osmosis happening, where water will keep coming into the cell. And so it's a way, if I make myself saltier than what's out around me, I can keep bringing water in, because water wants to, you know, dilute out all that salt or solutes. So that's one interesting thing that they can do. And then they also can actively pump out. If they get too salty, they can actually pump out and expend energy to do it, but they will pump out their solutes in order to make sure that they can stay alive and have enough water. High pressure, of course, it's the barophilic idea. Okay, so spend some time thinking about these ideas. Microbes are really quite flexible and adaptable, much more so than we are, to these types of conditions. And then temperature. Here they're looking at, this is kind of a classic graph looking at how temperature can impact pretty much any living organism. It's not just bacteria. But do you see how it's not like this perfect bell curve? It's skewed, right? You have this sloping off to the right. So as on the x-axis is temperature, and on the y-axis is growth rate. And it's not this perfect bell curve. So what that means is at colder, really cold temperatures, you know, organisms that aren't adapted to the extreme cold, what tends to happen then is the membranes gel up. And once your cell membrane gels and becomes more rigid, things get really challenging to the point of death, because your transport processes really suffer. Then you can see as you work your way warmer and warmer temps, you get some, you know, it's good, things are good. And then you get an optimum growth at an optimum temperature. So like with us, 98.6 degrees Fahrenheit, that's our optimum temperature. If we go below that, we can survive to a point, but eventually we're going to die. However, if you go slightly above that, so I don't know, I haven't talked to a medical doctor in a while about what's the death fever, what, 108 maybe, 108 degrees Fahrenheit, might, at sustained periods, would kill you. However, you can be chilled. We've heard of stories of people being chilled for hours and hours, and they can be resuscitated. And so this pertains to us too. It's not just microbes. But you can see that just a little bit beyond your optimum temperature, things quickly go bad to the point of death. And that's mainly, they think mainly because it's hitting the protein, the amino acid chains, and denaturing them. And if obviously proteins are so incredibly important for us and any living organism, if you start messing with that and denaturing the proteins, you're going to have some problems. Is there another chat, or why did that come up? Maybe that was me, I don't know. So typical range for a microbe, about 30 degrees, but it's skewed. So there's like a 30-degree range. So for example, E. coli likes 37 degrees Celsius. That's human body temperature in Celsius, 37. But they can only go a little bit above that, but they can go way below that, roughly to a 30-degree range. All right. So just keep in mind, it's skewed. Getting too hot is the big problem for most living organisms. So now if you think about the different categories of temperature adaptations, and we've seen some of these already, So I would like you to know these terms, please. Psychrophile, again, really cold conditions. And I'm not going to ask you to give me specific temperature ranges. Again, that's easy to look up, and it can vary depending on who's talking. So I'm not so concerned about that, but if you can put them in order, like, psychrophiles are like the coldest. They're well adapted to that, whereas the hypothermophiles are the hottest, all right, and then be able to map that out. We're mesophiles. We're that type of organism, and of course the microbes that hang out with us are going to be mesophiles. And then just above that you have thermophile. They are adapted to heat. Bacillus stereothermophilus is a fun one to work with in the lab because you have to grow them up. They actually like around 65 degrees Celsius. It's interesting to work with them. And then you have the hypothermophiles. And then usually they do, you see 2 hypothermophiles here because the last one is considered an extreme hypothermophile. So the next few slides are more just for study purposes. And again, I'm more concerned that you can put them in order, not memorizing exact temperature numbers. So don't get too hung up on that. It's just more of, do you understand what a psychrophile means compared to a mesophile compared to a thermophile? So these are just for you to kind of orient your head to these ideas, but don't get caught up in all the numbers, okay? All right, what else? Some ideas of where they live: volcanic activity, hot springs, hydrothermal vents. But even your backyard, you can have extremophiles showing up there. So that's one thing. I want to make sure you don't get caught up in thinking that they can only be in these areas where we would die. Not true. Compost piles in your backyard, if you get into the dead center of a compost pile, especially if you haven't turned it and mixed it in a while, it can get really hot. So it's impressive. And so you definitely know that you have extremophiles popping up in that area. Even a hot soil, because soil is so dark, all the tannins and the dark pigments, they can really absorb a lot of heat energy. So don't get too caught up in, yes, we can find them in really, volcanoes. Certainly, there are bizarre microbes there, but some of them are found right in your backyard as well. And like I said, fungi love acid conditions. So anywhere you can think where there's acids, they'll be hanging out and be quite happy. We're neutrophiles, of course. And then alkalophile is higher pH. And here, just a visual, if you want to look at it visually. Oh no, this is salt, sorry. So this is halo. Anytime you see halo, it's salt. And so some examples, and Vibrio fischeri is a marine organism. Halobacterium salinarum is a really interesting one. It's so easy to work with in the lab because you don't even have to autoclave the petri, the agar that you grow it on, because it's so salty, nothing else can survive. That's in your lab anyways, so it's fun stuff. Okay, so that's kind of the overview idea. And now as we work through the rest of the semester, I'll be touching back on this idea of the habitats, but that's kind of a general overview. We'll stop there. And next time we're going to get into sampling. So today's Wednesday, right? So tomorrow we do a Zoom lecture for everybody, and we'll get into methods of sampling, which I want to make sure to do before you get into your research project. Did you say something? There's a chat there. I just found out that when I end Zoom, the chat gets saved. I didn't realize that. So I will be checking the chats that way too. So if I miss it during lecture, and I apologize if sometimes I miss your chats, I will look them up after it's saved on my hard drive, and then I can answer any chats that way. So I might be sending out an email, but I'll get to it. Do I know when the test will be graded? Not exactly. I'm still picking away. I got 2 sets of exams to grade for both of my classes, and I'm almost done with the other one. So then I will be finishing up this one, certainly before the next exam, because you have to get the results back, and we have to go through it before I can give you the next exam. So I'm working on it. I know, I'm sorry it's taking a while, but essay questions take a while to grade. So I will be going over those as soon as possible with you when they're done.